It's fine. Okay. Okay, thank you, Denison, introducing me. And good afternoon, everyone. I am Hirofumi Ichinose from Kyushu University. So I'm very excited to be here because this is my first trip to Indonesia. Yes, I now understand no good food, nice sea, nice sky, kind people. Everything I need is actually here. So yes, so I'm also happy to be here to introduce our research topics in this kind of meetings. So I'm not happy if you could interested in about the Cytochrome P450 especially. And the topics for me today is biotechnological applications of fungal Cytochrome P450 Monooxygenases. I'm gonna include some topics, of course, in Cytochrome P450, but as also I want to introduce about natural products, especially focusing on terpenoids. And beginning of this lecture, not working. Guys are not working. We are sorry, please waiting. Okay. Uh, Thank you very much. So I would like to introduce about my research career at the beginning of this seminar. So I received PhD in 2002 in agriculture from Kyusu University. My main research topics I was working on the application of basidiomycetes. The basidiomycete is actually a English, but it's kind of scientific terms. So you don't need to understand basidiomycete. It means the mushrooms. Okay. And after I received the PhD, I got a position and postdoctoral position at Faculty of Engineering, Kyushu University. So I spent almost 3.5 years for enzyme engineering and nucleotide chemistry. And after postdoc, I got a permanent job at the Faculty of Agriculture, Kyushu University as assistant professor. Since then, I continue to work on the fungal biotechnology, including Bacillium, I see. But of course, we are using another fungal strains. And then I promoted to the associate professor in 2015, and I continue to do work on the fungal fungal biotechnology. Okay. Anyway, fungi is actually belong to the microorganism, but so there are three major kingdoms of living organisms. So, the bacterium kingdom, the archaea kingdom, and eukaryote kingdom. So fungi actually belong to the microorganism, but they belong to the eukaryote. It's the same as the animals and the plants. So fungi belong to the eukaryote kingdom. They should have a highly diversified metabolic system. So I could say that the fungal metabolic system should be much more complicated comparing to bacteria or archaea system. So many researchers are working on to utilize the metabolic system of fungi for SDS. So this is not my picture, but I strongly agree this figure because fungi are promising to achieve SDS because the actually mushroom is a fungi, but the mushroom is also food, isn't it? So biotechnology of mushroom is coming up with the zero fungus. And also some microbes such as Aspergillus species sometimes produce bioactive compounds. So good health and well-being is also coming up with the fungal biotechnology. But every goal are important for us, but I couldn't do everything by myself. So I am doing fungal biotechnology, especially focusing on the industry and innovation area. So today's topic for me today is only focusing on the fungal biotechnology, focusing on this industry and innovation areas. And so, but anyway, how many fungi are living on the Earth? It's very difficult to exactly answer, but it's estimated that 2.2 to 3.8 million fungal species should be existed on the Earth, meaning we, as humans, are living with a lot of fungal strains on the Earth. And the fungi are usually divided into 5 divisions called Ascomycota, Basidiomycota, Cryomycota, Zygomycota, Pseudomycota. So many fungi are living on the earth, and these fungi have a great impact on the human life, both positively and negatively. So positive impact may be fermentation technology. So you produce a tempeh in Indonesia, the fermentation food. It's made from the soybean, right? And you are using Dichomycetes fungi. Also, we Japanese produce fermentation foods called miso. It's a kind of soybean paste. Soybean paste miso is also produced from soybean, but we Japanese are using Ascomycetes fungi. You Indonesian and we Japanese both are producing fermentation food from the soybean, but we are using different fungal strain. It's very interesting. And also, on the other hand, negative impacts also sometimes happen because some fungal strain produce a toxic compound called mycotoxin that sometimes called a serious human problem called the mycotoxic diseases. And also, fungi is responsible for degradation of pollutants and carbon cycles in biospheres. So many fungi we should focus on, but I wanted to focus on Basidiomycota in this lecture especially. So Basidiomyces means mushroom, but why mushrooms are important for us? Because mushroom is able to degrade woody biomass. Wood is the most abundant biomass on the earth that is consisted with cellulose and hemicellulose and the lignin. The mushroom is able to degrade these organic components completely into the carbon dioxide water. That means the mushroom, Basidiomyces, are responsible for carbon cycle in biospheres. So that is why mushrooms are important for us. But anyway, we have 2 types of Basidiomyces called white rot Basidiomyces and brown rot Basidiomyces. So you can see here, whitish mushrooms here, brownish mushroom here. So which one is white? Which one is brown? If you think the white body mushroom is white, brown rot body is brown, it's not correct. Both are white rot Basidiomyces. The white rot and the brown rot does not mean of the color of mushroom body, but it means the color of decayed wood. White rot Basidiomyces decayed woody biomass into whitish colors. On the other hand, the brown rot Basidiomyces decay wood in brown colors. But why these color difference happen? The answer is the ability of Basidiomyces, whether they can degrade lignin or not. Only white rot Basidiomyces are able to degrade lignin. Of course, white rot Basidiomyces degrade cellulose and hemicellulose, but also lignin is completely degraded by white rot bacillomycetes. On the other hand, the brown rot bacillomycetes only degrade cellulose and hemicellulose, and brown rots do not attack the lignin. The lignin still remain in the decayed wood. Of course, lignin is a polymeric, aromatic polymer that can sometimes display brownish color if when the lignin is converted into quinone compounds. The quinone is orangish or brownish color. So the brown is come from the remained lignins. But so anyway, the ability of lignin degradation of white lactobacillomyces is very interesting. I can show you one general scheme of lignin degradation by bacillomyces. How bacillomyces, white lactobacillomyces degrade lignin. So bacillomyces, lignin is actually heterogeneous polymer. It's a polymer and a heterogeneous polymers. The first bacillomyces, it produces extracellular enzymes like lignin peroxidases and the manganese peroxidases and lactases. These extracellular enzymes initially attack the lignin to decompose its polymeric structure by non-specific one electron oxidation reaction. Because this one electron oxidation proceeded with radical reaction, during the lignin degradation processes, a lot of aromatic fragments, a variety aromatic fragments can be appeared from this process. And then fungi uptake all of these compounds into the cells and finally convert it into the carbon dioxide water. So lignin degradation by extracellular enzymes is very interesting. I want to show you one example of lignin degrading enzyme, lignin peroxidase. So lignin peroxidase is one of the most major lignin degrading enzymes produced by mushrooms, but it's a kind of heme-containing heme-containing peroxidase that characterize the general peroxidase cycles. The lignin peroxidases react with hydrogen peroxide to form active intermediate compound 1. This active intermediate oxidizes substrate 2 times. This reaction is almost a general peroxidase reaction. But the interesting point of lignin peroxidase is the lignin peroxidase have a high redox potential. Usually, peroxidase, well-known peroxidase, like a wholesale-based peroxidase, they only characterize one electron oxidation of phenolic compounds because phenolic compounds do not have a high redox potential. They do not characterize one electron oxidation of non-phenolic compound. In contrast, lignin peroxidase is able to characterize one electron oxidation of non-phenolic compounds because the lignin peroxidase have a high redox potential. For example, tetramethoxybenzene, lignin peroxidase characterize one electron oxidation of this compound to form allyl cation radicals. This allyl cation radicals undergoes to the quinone compound spontaneously. This strong oxidation activity of lignin peroxidase is very important to degrade lignin. And also, the strong activity of lignin peroxidase is able to utilize for the degradation of recalcitrant pollutants like dioxin. Dioxin is also oxidized by the lignin peroxidase to form the cation radicals. And these cation radicals undergo to the quinone compounds. They form quinone compounds, less toxic compound compared to the starting materials, dioxins. So many researchers are focusing on this extracellular enzyme system and apply for variety of application study, for example, bioremediation. So bacillomycetes are able to degrade a wide variety of chemicals. For example, sulfur contaminants here, it's found in fossil fuels and causing acid rain. So bacillomycetes are able to degrade these compounds. And of course, and bacillomycetes are able to degrade the chemical weapons called epirate, And pesticide here and the protein disruptor can be degraded by bacillomyces. So during the degradation process by bacillomyces of these compounds, the ligninolytic enzyme like lignin peroxidase or manganese peroxidase and lactases are sometimes important, play important roles, but not perfect because other enzyme system, not ligninolytic enzyme is one of most important enzyme, but the other enzyme also play important roles for degradation of these compounds. We also investigated this kind of experiments and we showed that the bacillomyces convert these compounds into the oxygenated metabolites. This oxygenation reaction was what were not characterized by lignin peroxidase, manganese peroxidase, lignin peroxidase. Extracellular one electron oxidation enzymes do not characterize oxygen transfer reaction shown in here. And we further investigated the possible involvement of enzyme system and sorry, we proposed that Cytochrome P450 enzyme system may play important roles in the degradation of chemicals by bacillomyces. So the extracellular enzymes like lignin peroxidase, manganese peroxidase, lactase attack polymeric lignin extracellularly. On the other hand, a cytochrome P450 system that I mentioned is intracellular enzymes that may responsible for the metabolism of aromatic fragments in the cells to the carbon dioxide waters. But what is cytochrome P450? The P450 is a kind of monooxygenase. The interesting point of P450s are widely distributed in nature and highly diversified in living organisms. To date, we could find more than 15,000 P450 genes in genomic database, but all of these P450 genes have been evolved from the single ancestor of P450 gene during the evolution of each organism. Because the molecular diversification of P450 genes in this process were achieved during the evolution of the organism, animals have animal-specific or animal-unique P450 system in their genomes, and also fungi also have fungal-specific or fungal-unique P450 genes in their genome. And plant bacteria, archaea also their own specific P450 system in their genome. And yes, and also cytochrome P450 is a heme-containing monooxygenase that characterizes the oxygen transfer reaction using NADPH and molecular oxygen. Single oxygen atoms is transferred to the substrate using NADPH to form the hydroxylated products. The interesting thing of P450 is that they are widely distributed in nature. For example, animals, we utilize the P450 for detoxification of drug and synthesis of human or animal hormones. So when you take a catch a course, you take a medicine. So all of the medicine should be eliminated from our body. So during the elimination of drugs, is characterized by P450 system in our livers. And plants also utilize biosynthesis, P450s for biosynthesis, so plant toxins, hormones, and flower pigments, and also fungi utilize the P450 systems for mycotoxin biosynthetic pathway. So like this, the P450s accept variety of substrate and they are playing variety of biological roles. That means the P450 system should be key enzyme system for the biotech biodiversity. And so P450 system is of course important to better understand the biochemistry, but the P450s are also interested in the biotechnology sector. This is the one success story of P450s. So this compound is a cholesterol utilized for treatment in osteoporosis. This medical drug, cholesterol, is produced from the vitamin D3 using two different P450s. Each P450 transfer oxygen atom here and here to form this medical compounds in one pot reaction. If you want to synthesize this compound in organic chemistry, you have to utilize the cholesterol as a substrate, but you also need 17 steps of organic synthesis steps. So tedious reaction steps needed if you do not utilize the P450. So like this, if we could find interesting and potential P450 system from nature, we can utilize for them in the industrial sectors. But a problem is what kind of organism is interesting source to find out the P450s, because animal have animal specific P450, plant also plant specific P450. Which one is a good candidate to harvesting the P450? And this slide shows the gene numbers of P450 in their genome. In case of animal, humans have 57 P450 genes in our genome. And house rat, it's an insect. Insects have a lot of P450, 90 P450 in their genome. Number is much higher than animal. And in case of a plants are rich source of P450, they have 250, and rice have 458 P450 genes in their genome. And also, fungal strain, white Lactobacillium, the phenylochiatic rhizosporium have about 150 P450 genes, and the brown rot Bacillium have 254. And the koji mold, it's utilized for miso production, 142, but the yeast is a very rare case, only 3 found in their genome. So gene number, like gene numbers, when we think about diversity of P450, gene numbers are actually important, but more important point is we have to think about the diversity of P450 family. The P450 family, so P450s are classified into the family depending on sequence identity. Two P450 share more than 40% amino acids. These two P450 categorized into same P450 family. So because the P450 family is this, P450s are classified into the family depending on the sequence identity. Diversity of P450 family means the diversity of P450 sequences. So please think that sequence diversity gives structural diversity, isn't it? Structural diversity gives functional diversity. That means the diversity of P450 function is very important to understand the functional diversity of P450s. So as you can see here, the fungi have a rich P450 family comparing to the other organisms like animal, plants, okay? Animal and the plants, bacteria. So meaning the fungal strain, fungi is actually rich source of interesting P450 functions. And this slide also shows the yes, this slide shows the phylogenetic relationships of fungal P450s. I constructed the fungal P450 phylogenetic tree of P450s using the P450s from two different fungal strains. The Blue one is a phanerochaetic lysosporium, white lactobacillomycetes, and red one is from the P450 is from posterior placenta. It's a brown lactobacillomycetes. And brown, bluish color indicate the PC specific clusters and or PP dominant cluster. And reddish one is the PP specific and the PP dominant cluster. So you can, as you can see here, so most of the fungal P450 share their own or dominant cluster. And also I compare the family diversity or in these two fungi. In case of PP, the PP have a largest P450 family belong to 5348, but interestingly, no P450 genes belong this family was not found from the PC. PC do not have P450 family. P450 genes belong to this family, only PP. In contrast, the PC have a largest family as 5144, and only few P450 genes belong to this family is from PP strain. PC have a lot of P450 genes belong 5144, but PP only have a small number of the P450 genes. So like this, we can understand that the molecular evolution of P450 genes have been achieved after speciation of bacillomyces rapidly. Of course, different evolutionary history should be involved in the diversification of fungal P450s. So it's very interesting gather a lot of P450s from different fungal strains. So to better understand the catalytic function of P450s, so we try to construct a functional library of P450s. So it's not so difficult. It's not easy, but it's not so difficult to find the P450 genes because now thousand fungal genome projects is now going on, and more than genomic sequences of more than 600 bacillomycetes is now available on database. We can easily access to the genomic database of bacillomycetes genome, but problem is that the sequence information does not tell us their catalytic function. We can easily access to the P450 sequences from the genomic database, but it's not difficult. It's not easy, and it's always difficult to estimate their catalytic function. So to better know their function, we then try to construct a functional library of P450s. We first try to identify P450 genes from genomic database. So gene candidates could be easily find from genomic database, and then we try to isolate the cDNA from fungal mycelium by RT-PCR, and then clone into expression vectors. This expression vector is available with the each strain Saccharomyces cerevisiae. Each of the P450 genes were cloned in expression vectors and then transfer into the Saccharomyces cerevisiae. Saccharomyces cerevisiae, the transformance of Saccharomyces is able to express the fungal bacillomyces P450. Then we added substrate into the transformance and to do the bioconversion experiments. During this kind of experiments, the first battle was actually cDNA isolation. So because we wanted to isolate many P450, as many as possible, we tried to extract RNA that from the fungi that were cultivated in different culture conditions and for different ages. And once isolated cDNA was then utilized for the heterologous expression or in Saccharomyces cerevisiae. The East strain Saccharomyces is a very good expression system to express the fungal Bacillomycetes P450s, we confirmed that more than 57% of P450s could be expressed, heterologously expressed in Saccharomyces cerevisiae. So yes, actually we spent more than 10 years to isolate the cDNA. Now we have 615 P450 genes as the full-length cDNA. This is the most largest P450 library in the world. So we have 615 P450 genes, and all of them were transformed into East strain, meaning we have 615 East transformants that express different P450s. So using these transformants, we can do screening using a variety of substrate. Yes, using this system, we published many papers and did conducted collaboration work with Japanese company or German companies. So, but I want to show you some example. So this is our original example. So we utilize the genistein for screening. Genistein is the abundant natural products found in soybean. So genistein, well, when genistein was incubated with transformants that express CYP57B3, so metabolic products were detected on HPLC, and we identified these metabolites as 3-hydroxylated or 6-hydroxylated, the 3' hydroxylated genistein. So interestingly, the metabolic products have been suggested that they have antioxidative activity or anti-HIV activity or anti-cancer activity. This bioactivity could not be found in starting material genistein, meaning we could produce the value-added natural products from naturally abundant soybeans, isoflavonoids, using P450s. Okay. And also we try to improve their catalytic function using mutation technologies. So because wild type, actually the wild type of CYP57 convert the genistein, but its catalytic activity is not good. So to improve the catalytic activities, we created the mutants using a random mutation technique. So successfully mutants showed good activity with high kinetic parameters comparing the wild type. So we could improve their catalytic activity to achieve more effective conversion. And also, this is another example. The abiotic acid is found in the pine resin. It's also naturally abundant natural product. And abiotin is converted by CYP5012N3 to rare natural compounds. We are not sure whether this rare natural compound have bioactivity or not, but we could say that the abiotic acid is actually converted to the rare natural compounds using a fungal P450 system. And also other P450s produce the metabolite A and the metabolite B, C, D. These compounds are different from this one. So we could produce a variety of rare natural compounds using P450. And another example is the testosterone. Testosterone is the animal steroid, right? But interestingly, the fungal P450s, many fungal P450s able to combat testosterone, even though fungi do not produce testosterone. Okay. Yes. And also testosterone conversion by the Salmonellium P450 have been successfully identified by Dr. Dani-san. Dr. Dani identified the testosterone 40 alpha hydroxylase from Salmonellium elegans. The 40 alpha position, oxidation of oxygen transfer reaction at 40 alpha position, I'm sorry. So slide is not okay. The oxidation at this position is very difficult due to the strike hindrance, but among the 615 P450, only this P450 catalyzed the chlorine alpha hydroxylation reaction. This study was achieved by collaborative work with Bayer from Germany. So yes, we have a lot of experiences. A lot variety of chemicals can be converted by the fungal P450s, even though I only show some example in this seminar, but we have some experiences on steroid compounds and industrial chemicals that sometimes cause environmental pollutants or plant-related compounds of pharmaceutical compounds. Many compounds can be a candidate of a substrate. And yes, so functional library gives a direct transfer. As I mentioned before, it's very still difficult to estimate the catalytic function from genomics database and their sequence. But we could directly evaluate the catalytic function if using the P450 functional library. Of course, we welcome your potential substrate. We can do screening with your substrate if you could provide. Okay. Okay, here I want to switch another topics. So I said introduce about our P450 system. Actually, the P450 is our library. It's a very powerful way to screen out the catalytic function. And also we try to extend this kind of experiment to the rational synthesis of natural compounds. So there are a lot of natural compounds in nature, but my target is mainly focusing on the terpenoids. Terpenoid is the largest class of natural products produced by plants and microbes. They are synthesized from DMAPP and IPP as an intermediate through the intermediate formation of some kind of intermediate here. And terpenoids are usually divided into some class depending on the carbon number. For example, C10 compounds, the monoterpenoids, and C15 sesqui, C12 diterpenoid, C30 terpenoids. In this seminar, I want to only focus on the sesqui terpenoids here. So sesquiterpenoids are very interesting terpenoids because they show the fascinating diversity of chemical structure. And sometimes they show the interesting bioactivity. For example, iridine S, it's produced from poisonous mushroom, but this compound shows anti-tumor activity. And also, I think the most famous sesquiterpenoid in the world is artemisinin. Artemisinin is produced from the plants and it's have anti-malarial activity. And the bisabolol, it's produced from herbal plant and it also shows the anti-irritation activity like this. So many sesquiterpenoid compounds have a biological activity. It's going to be a good candidate for pharmaceutical application. And so this slide shows the general scheme of sesquiterpenoid biosynthesis. There are a lot of sesquiterpenoids in nature, but all of the sesquiterpenoids is produced from the common ancestor, a common intermediate, farnesyl phosphate. This intermediate is converted into the cyclic compound. It's a scaffold of compounds by the action of STS. And then these scaffold compounds was further converted into the complicated chemical natural compound using a degradation enzyme like Cytochrome P450s or other enzymes. So because we have a lot of P450 in our laboratory, we library the P450, but we didn't have SDS genes. So if we could utilize both SDS and the P450, we could do the rational synthesis of sesquiterpenoids in laboratory. So we do try to rational synthesis of sesquiterpenoid, we first to isolate STS genes from the genome using a genomic database. As I mentioned before in P450 experiments, so it's not difficult. Yes, it's not difficult to identify STS genes from genomic database because more than 600 genomes of bacillomycetes are available. Easy to find STS genes. To isolate the cDNA of STS, we selected 7 bacillomycete species based on the selection criteria that fungi are rapid growth in synthetic culture medium, and some of them are edible mushroom, some are medicinal, and well-studied mushroom have been utilized in this experiment. So from these 7 bacillomycetes, we could isolate a lot of cDNA as shown here. First, we identify gene candidates from genomic database. So we identified 122 candidates and experimentally I isolated 71 cDNA from the 7 bacillomycete species. And the isolated cDNA of STS was then transformed into Saccharomyces cerevisiae to express XSTS. The metabolites produced by STS are both accumulated in headspace or culture medium. So we isolated the metabolites from both headspace and culture medium, then analyzed by GCMS. So yes, at least, so 40 scaffolds were putatively identified by GCMS analysis. You can see here so complicated chemicals appeared by the action of STS. Now we can produce a highly complicated sesquiterpenoid scaffold using STS or bacillomycete. And then we further analyze the phylogenetic relationship and catalytic function of STS. This slide shows the phylogenetic tree of STS constructed based on the sequence similarity. When STS from the bacillomycetes are liable to be classified into 5 class. And when we look STS from class 1 and 2, so these STS liable to produce a variety of metabolites. That means the product's specificity is not so high when the STS is belong to class 1 and 2. In the contrast, the STS classified into 3, 4, 5 produce single major sesquiterpenoid scaffold. It's a bit different feature when we compare class 1, 2, or 3, 4, 5. And so, yes, during these experiments, we successfully identified the novel sesquiterpenoid scaffold and named Preostin. The Preostin was synthesized by the STS from Pleurotus ostrotus. Pleurotus ostrotus is a very famous and popular edible mushroom in Japan. And we could identify the new scaffold compounds from this edible mushroom and chemical structure confirmed by NMR spectrum. Furthermore, we confirmed the absolute configuration using crystalline sponge method. The crystalline sponge method is a powerful way to understand the absolute configuration. So finally, we established the chemical structure shown here and released named Preostin. Yes, the finding of Preostin, it's a new compound. It's very interesting for us and excited too much. But more important thing is that this new compound was identified from Pleurotus ostrotus, very common edible mushroom. So many researchers have been working on this edible mushroom. Of course, basic study, application study, many studies have been conducted using Proteus austratus. But we identify new compound from this one, meaning so we know only a little about the diversity of sesquiterpenoids in Bacillomycetes. So the protein is actually belong to Ariacan type sesquiterpenoids. When we look Ariacan type sesquiterpenoids, some researchers have been reported and isolated Ariacan type sesquiterpenoids from Bacillomycetes, and some of them show the anti-tumor activity, antibacterial, antiviral activity. We were not sure whether the protein or Proteus austratus produce this kind of bioactive sesquiterpenoids or not, but our finding should be useful to better understand the naturally occurred Ariacan type sesquiterpenoids. And also another example is delta 6 protoidodine. Delta 6 protoidodine is considered to be the most possible scaffold compounds of bioactive compounds meleoride or amylyl oxalide produced by Aminaria species. So we identified the protein from posterior placenta. We were not sure whether posterior placenta also able to produce this kind of metabolites or not, but we were lucky because we have a lot of P450s from this one unit. We have already isolated 184 isoform of posterior placenta P450s that cover 75% of total P450 encoded in this fungi, meaning we could conduct a semi-comprehensive screening using 184 isoform of P450. So to screen out the P450 that characterize there's a 6 protoidodine hydroxylation, we constructed the whole expression system of STS and the P450 using two different vectors. Two different vectors was co-transformed into Saccharomyces to express both P450 and STS. Based on the comprehensive screening, we identify the CYP5344 that characterize delta 6 protoidodine 8 hydroxylation reaction. And this product is not so much stable in acidic culture medium, because this compound is spontaneously converted into the delta 7 protoidodine 6-ol R form and S form. This is not enzymatic reaction, spontaneously occurred. And also CYP5348E1 characterize hydroxylation at a different point, 5-ol to produce 5-ol. And CYP5348J3 also characterize hydroxylation reaction of delta 6 protoidodine, even though we could not identify the chemical structure of metabolites. So based on this study, we could figure out the possible metabolic pathway of delta 6 protoidodine metabolism by posterior placenta. The posterior placenta first produce delta 6 protoidodine using a STS08 and then convert it into 8-O or 5-O or other hydroxylated products using CYP5344 and 48E1, 48J3. Yes, so we are still thinking that this compound is true intermediate and posterior placenta may produce highly complicated natural products using this compound as an intermediate. So we are now doing checks the further enzymes involved in the production of natural products. Yes, like this, the better understanding of natural products of sesquiterpenoids. So studies on SDS and the P450s are, of course, important to better understand the natural product. But in such case, we have to do experiment with the natural combination. For example, SDS from posterior placenta and SDS from P450s from posterior placenta. But what's going to happen if we combine together with non-natural combinations, for example, SDS from edible mushroom and the P450s from poisonous mushroom? It might be possible that the pseudo-natural, not natural, pseudo-natural sesquiterpenoid could be appeared with by the non-natural combination of SDS and P450. Yes, we have a lot of P450s and a lot of SDS. We could combine together, even though they are non-natural combination. Yeah. So to achieve the conversion of sesquiterpenoid scaffold by the non-natural P450, we constructed the co-expression system using 5 species of SDS and 115 species of P450, meaning we created more than 3,000 P450 transformers that express SDS and P450. The number of this is not actually correct because my student is working on day by day. That number is now increasing day by day. I guess more than 10,000 transformers have been constructed. So I want to show you one example of the non-natural combination here. So this is metabolites produced by the action of SDS from Tramatis vescera that produce a gamma catenane synthesis. And this SDS was combined together with P450s from phenylochiated chrysosporium. This is a metabolites detected on GCMS, and we identify the 3-alpha hydroxy gamma catenane synthesis. Gamma catenane was produced by the action of both Tramatis SDS and phenylochiated P450, meaning the first intermediate was cyclized into the gamma catenane by the action of Tramatis SDS, and then this one was further hydroxylated that one of P450 from phenylochiated. So interesting point is that the phenylochiated does not process gamma catenane synthesis. This gamma catenane synthesis could not found in phenylochiated, but phenylochiated P450s is able to catalyze conversion of gamma catenane. This is actually, we could successfully achieve the non-natural combination pathway here. So using this non-natural combination of SDS and P450, it might be possible to produce a rare natural or pseudo-natural compound in laboratory. And also, this is another example that we utilize the proeostem. The proeostem synthesis was identified from edible mushroom, Prorotus osculatus. The proeostem was converted into this compound, even though the structure was still proposed structure. But this proeostem was actually effectively converted by this compound. And this P450 is from Salmonidum P. elegans that belong to Zygomyces P450. It's all completely different fungal phyla, meaning that we usually synthesize these compounds using Bacillomyces SDS and Zygomyces P450. Completely different fungal enzymes should be combined together. Yes, this is a final slide. Yes, we now trying to produce a variety of non-natural or pseudo-natural sesquiterpenoids. So we have achieved to produce this number of sesquiterpenoids. So now we are checking out the bioactivity of this compound. Yes, we hope that we could find out interesting bioactivity in this compound, but we were not sure whether they have interesting or not. But actually, we could say we could produce rare or non-natural substrate using SDS and the P450 isolated from the variety of fungal strain. Okay. Okay. So this is my final slide. So Fungal P450. Yes, I showed the Fungal P450 showed molecular and functional diversity, and it's still challenging to figure out their catalytic potential, but we can now utilize the P450 library to find out their potential functions. And experimental approaches tell us unexpected functions of P450s. And also I presented the diversity of sesquiterpenoid of basidiomycetes, and we say that the basidiomycetes are rich source of sesquiterpenoids, and fungal P450s allow us to produce a variety of rare natural, pseudo-natural, non-natural sesquiterpenoids in laboratory. So we are now checking out the more potential bioactivity of this kind of sesquiterpenoids in now. Okay, that's all from my side. Thank you very much for your kind attention.
Thank you very much for Ichinose Sensei for your impressive and interesting presentation. So now we are going to start the discussion session. So if you have some questions, just raise your hand and mention your name, tell us your name and then what the question. Okay. We will start from online participants. Okay, there's some question from Nofianto, Mr. Nofianto from Malang. Okay, how does the creation of mutants, fungi strains, and microbial engineering, especially fungi like Corsia, difficult? Yeah, something. In the industrialization of biofertilizer and biopesticide that have high efficacy. And second, it's okay for you? You can answer one by one or you can answer. Okay.
Can I see first one? First question. Yes, I want to ask some question to how does the creation of mutant fungi train and microengineering under like that's what it is. How does me? How does yes we constructed the mutants based on using the random mutation techniques because of course actually the rational mutation is sometimes possible, but I guess it's still difficult in the case of P450 because the P450 substrate specificity of P450 is not so easy to understand because the P450 accommodates substrate in the cavity of the hem. The structure of the cavity of hem is determining the substrate specificity. It's not difficult to control the shape of substrate specificity rationally. So we try to create the mutants using random mutation technique. And in number two, number two where? Number two. What the potential endophytic fungi engine, endophytic means? Producing plants. The potential endophytic fungi. secondary metabolite form. Scale using bioreactor. Yes. Yes. Genomic engineering or fungi, of course, we are interested in, but it's quite difficult, still quite difficult because yes, now, like a CRISPR-Cas9 genome editing technology is growing up, but it's still difficult in case of fungi. And also, plasmid that can be utilized for in the fungi are only limited. We have to do the genome integration of some genes. So we can utilize plasmid DNA if we use the yeast Saccharomyces, but if we utilize the filamentous fungi, in case of filamentous fungi, we have to achieve to create transformer based on the genome integration. So that is why it's still difficult to achieve the fungi engineering, endophytic fungi engineering. It's very interesting and the potential, but need more technology so far, I think. And
Next question, Jason.
three,
He want to ask you again.
What? see,
What? Edible mushroom breeding.
どこ
はい。
あ、これか。Post harvest technology, edible mushroom in Japan?
Yeah.
Post harvest technology of it. How the development cultivation techniques and yes, to isolate the possible secondary metabolites. Yes, yes, it's interesting to harvest secondary metabolites from the post-harvest technology. But yes, we were not sure that whether the mushroom actually cultivated in the industrial scale could produce actually this kind of active metabolites or not. We suggested the latent potential to produce active bioactive secondary metabolites, but it's not always true that mushroom product produced in industry always producing the secondary metabolites. Of course, it's interesting in the potential if it's possible, but we are not sure whether the bioactive secondary metabolites are always produced in the post-harvested mushroom or not. Good.
Any other question from the online participants? Okay, sure. Okay. Okay, how about the offline participants? Do you have some questions? Okay, please.
Thank you very much for the presentation. It's very interesting. My question is, if we want to test the biosubstrates, for example, we have several biosubstrates to test with the enzymes of the fungi. So how do we test it? Is it like we have to isolate the enzymes for the fungi and we test it in vitro? Yeah. And my second question, if the biosynthesis and the bioprocess, where do this process happens? Is it inside the fungi, or we have to like isolate the enzyme for the industrial purpose, or for the biosynthesis purpose? Usually the biosynthesis happens in the cells, right? Yeah, that's my question. How makes sense?
Thank you for your questions. So, Sensei.
Actually, we want to construct the bioprocess in vitro, like a bioreactor using a purified enzyme or immobilized enzyme, something like that. But I think it's still difficult in the case of P450 because the P450 is membrane bond enzyme. It's very difficult to harvest, to isolate in vitro and activate in vitro conditions. I think it's better to utilize the whole cell system to produce the secondary metabolites or not. And also P450 need NADPH to activate catalytic cycle. It's very expensive chemicals, but when we can utilize the whole cell system, NADPH is produced always when the fungi is growing up. So I think utilization of whole cell system or fungal system is much more practical to produce a natural products. Okay, did I answer for your question?
On your slide, about the. Yeah, I read in on your slide that you mentioned do you have any substrates to test? So I'm wondering, where do the testing happen? Is it like inside the fungi or in vitro outside the fungi?
So Sensei, I want to just ask, add some information. So she want to ask about the substrate. Is it possible to do the screening P450? Yes.
I say that all of the chemical that we have can be possible to screen out using a P450 screening system. But unfortunately, we only can detect on HPLC now in my lab. We utilize HPLC system. That means the chemical with high absorption, UV absorption, or can be detected on MS system, on GCMS, only possible. But basically, it's all chemicals can be possible to screen out. Of course, natural products or chemicals or pharmaceuticals can utilize for screening. Okay.
Is that enough for you? So maybe I will just add some information regarding my experience working with him. So if you have substrate, because Sensei has like 615 genes in library from 5 species of fungi, and we always use as whole cells biocatalyst. So the genes will be expressed in cells, and we do not do the protein expression and purification because, like as he said before, P450s are membrane-bound proteins. It is hard to be expressed and purified into the medium. So the substrate that can be applied as, let's say, precursor for your biosynthesis using P450s, you can use anything. Drugs. Last time we used drugs, and then other pharmaceutical compounds, and then organic compounds, as long as the substrate can be soluble in the water, because we use medium, and the medium, for yeast growth. And if the substrate is not soluble, we have to add some surfactants or BMS or others, but it still works if you want to use. Yes.
Now, is it possible to express it in the bacteria? Since, you need to extract the protein from the bacteria cell wall.
Yeah, I have tried before. We expressed some P450s using E. coli, but unfortunately very low expression level because they still membrane-bound protein, right? But there are 2 classes of P450s that, yes, still difficult, very low expression level, but When we try to characterize the expression level using the absorption at 450 nanometers, the expression level, the absorption was low, but when we use the biofunctional using substrate, it still work. So sometimes, although the expression levels are low, but it's still working to catalyze the bioconversion reaction. So that's the unique of P450, something like that. Yeah. Any other question? Oh, please.
Okay, hello, Ichinose Sensei. I'm Pradip from Brin. I do have a question. So you mentioned that this is the whole cell biocatalyzed, and then you use substrates of secondary metabolites. Sorry, my question is, can the fungus uptake the substrate naturally, or do you need to perform some kind of genetic modification, like specific uptake genes in order to perform the reaction? And then the second question, because this is oxidase enzyme, do you also need to co-express the reductase in order to maintain the redox balance inside the cell? Thank you.
Uptake of substrate
Into the cell.
or which one? Said SDA.
Yeah, cytochrome P450.
Yes, we didn't check exactly what kind of substrate chemicals can be uptake into the cell or not, but secondary metabolite. So, yeah, you try the co-expression systems of SDS and the P450s. Yes, SDS first work in the cells. Yeah, we don't add the scaffold compound before the reaction of P450, doing work together in the cell same time at same times. So I'm not sure whether the transformed scaffold compound once go to the outside cell or not. But actually, I guess both reaction achieved in intracellularly. Yes, this one, right? Yes, but actually we checked the metabolites using only this SDS only. We could isolate this compound from the extracellular fraction. So that meaning the once formed scaffold compound definitely going outside. But also we could achieve this degradation reaction using P450. That means the P450 quickly react with the scaffold compound after the formation of the scaffold compound.
Second question about the case.
Second question is,
Oh yeah. So this is Monooxygenase, right? But do you need to co-express reductase as well in order to maintain the redox balance?
expression of other enzyme.
Yeah,
reductase for P450 redox enzyme. Yes, good question. I think. Yes, we usually the P450 reaction set plasmid present. We usually the expression vector. Okay, これでいい。This is a scheme of expression plasmid. We usually the PGYR that have P450 genes, and it's also contain the CPR. It's a Cytochrome P450 reductase that donate a electron to the eukaryotic P450s. Yes, this one plasmid produce both P450 and its redox partner CPR heterogamously. If you remove this CPL, the reaction efficiency is not good. I think it's important to express the CPR for plasmid. Did I answer for your question?
Next question. Please, Miss Alifa, if you want to, yeah, you can switch your mic on. Okay, just
Thank you. Minasan konnichiwa, hajime masitu watashi wa
konnichiwa.
Hanjiku karakemashita, semijajar sudah imas, doso yoroshiku onegasaimasu. Excuse me, Prof. Hirofumi. Let me introduce myself. My name is Alifa Marita Gani Sabrina. You can call me Gabriel. I'm studying college from East Java, majoring in skin education. May I ask, what are the main application of P450 of fungi in the field of biotechnology? Thank you.
The
Okay.
main application of P450s.
Yes.
Yeah, the main application. What main
P450
application?
of fungi in the field of biotechnology?
Yes, your question is the main application of P450 technology, isn't it? I think the most interesting point of P450 is it's able to transfer hydroxy group into the variety of substrate that is site position specific or yes, specific reaction. Site specific or yes, site specific hydroxylation reaction or sterol specific hydroxylation reaction is very much difficult by the organic synthesis. But in case of if you could utilize the P450s for the hydroxylation of such difficult instead of such difficult organic reaction step. So we could produce a variety of complicated chemicals by biotechnology. So it reduce some the processes that should needed. We need organic chemicals or toxic catalysis or high pressure or environmentally non-friendly process should be changed to the P450 technique. We could produce a variety of chemicals that could not be achieved by organic synthesis processes. Variety of chemicals can be produced.
Is that answer your questions, or do you want to follow up your questions?
Yes.
Okay.
Thank you, Prof.
Okay. Is that enough for you?
Yes.
Okay. Thank you.
Okay.
So
Thank
we still have 14 minutes, or yeah, we can discuss anything. Okay. Dr. Enda, please.
So I want to ask, so far until recently, how many enzymes have been functionally elucidated in your laboratory? And secondly, I'm curious, if we send a substrate to your lab, so what will you do? I mean, which enzyme will you use to try to metabolize the substrate? Yeah. Thank you.
Substrate.
Yeah, Sensei. She asked about possibility to send some substrate, right? Yes. Now, Dr. Enda, just we'll use all 600. Yeah. So just for your information, Dr. Enda, currently ITB, School of Life Science and Technology and Chemistry Department, in collaboration with Ichinose Sensei, so we can send and or request a substrate or more than one, okay, and he will do the screening. So if you are also interested, you also can join with us, no problem. So Sensei, he asked about possibility to do screening. Yes.
Collaboration.
Yes, yes. Is it possible? Because and how many enzyme that we will use for
Yes,
screening?
collaboration is of course possible, but unfortunately we can provide the gene for your laboratory. It's only available in my lab. If you could propose some substrate or chemicals, so we could do screening in my laboratory and share the information, but we couldn't provide the gene itself without some contracts between the brain and university. My genes belong to the university. It's not home for me. But I think it's possible. It might be complicated. Not
Yeah, the same problem. Now we are facing same problem because we asked Ichino Sensei whether it's possible or not to send some plasmid to Indonesia to ITB, but unfortunately. So, and then he proposed if you have some substrate, interesting substrate or targeted substrate, you can just tell him and he will find the substrate in Japan, buy it, and will use for the screening. And as you may may aware that the enzyme is 615, so he will use all of them. And if you got the hit, the interesting result, you can more characterize further in Indonesia by constructing the synthetic gene and transform it to yeast, and then we can do further characterization in Indonesia, something like that. Yeah. So he is very open if all of you want to do the screening using your own chemicals or well-known chemicals like commercially available in Japan, you can just contact him. Yeah, something like that. Okay, any other questions? Okay. Please, I think you're still curious about his research, right?
450, it's similar with those in human, right? I mean, like human also have the P450. So yes, but we have like several families. So how many of this P450 and the fungi that have similar function as the humans, as those in humans? Thank you.
So Sensei, how many P450s are similar from between human and fungal? Something.
basically we couldn't find same P450 family in between fungi and animals. Only CYP51 is the same between the fungi and animal. 51 are responsible for the metabolism of steroid compound. In case of human, we utilize P450 to make cholesterol. And in case of fungi, they utilize CYP51 to make ergosterol. It's very similar compound. But yes, only this example. CYP51 is common in the animal, plant, and fungi, but other P450s are completely different sequences. No identical sequences can be found from both animals and fungi.
Thank you, Sensei, for your answer. Any other question, or maybe I will give you two more chance to ask him. Just for your information that this P450s also still not too attracted many research from Indonesia. When I try to briefly Googling P450 and Indonesia, no results. I mean, in terms of fungal P450s, many Indonesians only focus on human P450s for metabolism of drugs in the liver. So I think it's a chance for us. We study about fungal P450s because we are mega diversity. We have a lot of fungi, and in ICC, Indonesian Culture Collection, there are so many fungi, and we have to explore them with, whether they have fungal enzymes, fungal novel P450 from fungi, or something like that. There are many chances for us to explore the enzymes. Okay, I think there's no more questions, so I think I can close this session, or we will check the last check from the online participants. No? Okay. Okay. I would like to conclude our discussion. So first is the potential application of potential application of fungal P450s are promising, not only for bioremediation, but also for biotransformation. And as far as I understand, these P450s also can catalyze sulfoxidation reaction and ring opening reaction and some difficult reaction of organic synthesis. So if you are interested in this enzyme, is very welcome. Maybe for protein engineer or enzymology, this enzyme is kind of strange because the name like CYP number and something, because they have their own nomenclature. So CYP and then 5 or something belong to the family, subfamily, and then the species name. So as you can see here, like you cannot see like amylase with A's or others, but here you saw some numbers. Maybe it will make you like confused about the name of the enzymes. Second, Ichinose Sensei is open for collaboration. So in case you are interested to do some screening, just contact him and hopefully we can have collaboration in the future. And that's all from me. Please give your best applause for him. Thank you very much. That's all from me as a moderator. I will return it to the MC. Thank you.